Bismillah ar-Rahman Rahim. This is the A-Level Biology March 23 paper 2-2 and this is the second video. Question 1 and 2 have been done in the first video. We will probably do 3 and 4 in this. Question number 3a, figure 3.1 is a diagram of an area of the phloem tissue from a transverse section through the stem of a squash plant, whatever. So you can see there's X, there's a companion cell next to X, and then there is Y. So you've got to be really understanding cell X and cell Y in figure 3.1 are sieve tube elements. Explain why cell X and cell Y have very different appearances in the transverse section. Why the cell Y is showing you the sieve plate. If you look at something from the top, well, you're going to see the sieve plate and has been cut at a section. So cell Y shows the sieve plate or the sieve pores, and the sieve plates are at different heights. So they would be cut in a different section. The section misses the sieve plate in cell X. Now just look at two glasses like this. They both have water in them. Now if you look at a section of them, if you take a section from here, both will contain the water. But if you take a section from here, now you can see there's no water in this one, while here there is water in this one. So similarly in that question they're saying, okay, so there are different sieve tubes. Now this is a sieve tube, and this is a sieve plate, and there's another sieve tube here, and the sieve plate is here. So if you take a section from here, or if you take a section from here, or if you take a section from here, so in one you can see the sieve plate and the other you can't see the sieve plate. So cell Y shows a sieve plate or a sieve pores, and the sieve plates are different heights in the stem, and the section misses, or you could have said the section misses the sieve plate in the cell X. Now just to get you clear about this, I don't think some of you are very clear about it. Now you look at here, we have the sieve plate here, and here we have the sieve plate here in this one. So if I cut a section from here, then I probably do not get another sieve plate anywhere. So that is why the diagram that you see is something like this. Now these also have sieve plates, but they're not in the same level. They could be a little below or above or somewhere else. So you can see these, but you cannot see in the other ones. Then part 2 is a very direct question. Sucrose is formed from the glucose synthesized by mesophyll cells in the leaves of the sea. Peppo, explain how companion cells are involved in the transfer of sucrose into phloem sieve tubes. So you know this is the sieve tubes, and these have the sieve plates. Now here we have the companion cells. So the companion cells are here, and we have to understand how this is going to happen. So basically what is going to be happening is hydrogen ions are pumped out into the cell wall, and when they diffuse back in, they also bring along with them sucrose. So basically sucrose is co-transported into the companion cell. These are the companion cells which are next to the phloem, to the sieve tube. And then of course through the plasmodesmata they are going to enter the sieve tubes. So how do you word this? Oh, you word it very easy. It's hydrogen ions or protons in the companion cells are pumped out by active transport. This establishes a proton gradient. When the hydrogen ions are pumped out into the cell wall, I don't mean outside the cell, I mean into the cell wall, then the protons move down their diffusion gradient. They're going to pass through facilitated diffusion, but this is going to be a question of co-transport. So I send you to go and get some water from the canteen. You go and get water, but you get some juice for yourself as well. So co-transport, the sucrose is co-transport, moves against its concentration gradient through a co-transporter protein, which is also called as symporter, SYMPORTER. And the diffusion of sucrose will then occur into the phloem sieve tube through the plasmodesmata. So this was the plasmodesmata, this space. There is this space, this is called plasmodesmata, this area where there is a channel or there is a connection between this cell and this cell. So those are called plasmodesmata. So wording it, protein and companion cells move protons into cell wall. So there's an active pumping. Using ATP or by active transport, hydrogen ions pumped out of the cell, establishes a proton gradient. Protons move down their concentration gradient, co-transport with sucrose Sucrose moves against its concentration gradient through a co-transporter protein and the diffusion of sucrose into the phloem sieve tubes through the plasmodesmata. Very simple mark scheme. This has come in so many papers and everybody should have memorized this because you know this is going to come again and again. Hydrogen bonding is important in the movement of water in the xylem. Explain how hydrogen bonding occurs between two water molecules. And then outline how hydrogen bonding is involved in water transport in the xylem of a plant cell. So again, a very direct question. For part 1, you could have even given me a diagram of two water molecules showing the hydrogen bond. So basically you have to say that oxygen has a small negative charge and hydrogen has a small positive charge. Then the hydrogen bond forms between the oxygen of one water molecule and the hydrogen of a neighboring water molecule. Or you could have said oxygen is more electronegative than hydrogen. So you had to give me an explanation for that. Then coming to how hydrogen bonding is involved in water transport in the xylem, this part of the question. Cohesion between water molecules gives a column of water. Hydrogen bonds make the water column difficult to break. Cohesion, basically how is water going to move up is cohesion and adhesion. So you have to explain that to me. You just writing cohesion and adhesion would not get you any marks. So cohesion is between molecules, between water molecules giving you a column of water. Hydrogen bonds make the water column difficult to break because it's so tightly attached to each other. Then hydrogen bonding or adhesion of the water molecule to the cell wall. Then polar water molecules, hydrogen bond to the hydrophilic cellulose or parts of lignin. Then adhesion supports the column of water. Then water pulled upwards by transpiration or evaporation. So basically, you could have said how hydrogen bonding occurs between the water molecules. You have to give me an explanation. Oxygen small negative charge, hydrogen small positive charge. Hydrogen bonds form between the oxygen and one water and the hydrogen of the other water molecule. Or also you could have said oxygen is more electronegative than hydrogen. I'm just going to explain this to you in a diagram. Now, as you can see in this diagram, you see oxygen has a partial negative charge and the hydrogens have a partial positive charge. Now this of course results in, you can see how the oxygen and the hydrogen are going to bind, and these are the hydrogen bonds. And these hydrogen bonds are holding these water molecules together. Now this is called cohesion or cohesive forces. And because of this, a continuous column is formed. And it's difficult to break this continuous column because of these many hydrogen bonds. Another diagram showing you these hydrogen bonds and how these hydrogen bonds are going to form a very interesting structure by which you cannot break a column of water. So outline how hydrogen bonding is involved in water transport. So as I said before, cohesion between water molecules gives a column of water. Hydrogen bonds make the water column difficult to break. Hydrogen bonding or adhesion of water molecules to cell wall. So you have to learn these wordings because some of you are unable to put your thoughts into words, and that's because you see you don't really these days the present generation doesn't read a lot, so they are unable to express what they want to say. Cohesion between water molecules gives a column of water. Hydrogen bonds make the water column difficult to break. Adhesion of water molecules to cell wall. Adhesion supports the column of water. Water pulled up by transpiration, or you could have said evaporation. Hydrogen bonding between water molecules gives water a relatively high latent heat of vaporization. Suggests why it's important to plants that water has a high latent heat of vaporization. Now, how you have to think is what would have happened if it was a low latent heat of vaporization. So basically, high latent heat of vaporization means that it is going to take the heat. So it's going to have a larger cooling effect. Like for instance, when we sweat and the sweat evaporates, it cools the body down. But you produce very little sweat. And when that evaporates, it cools the body down. But if it was a low latent heat of vaporization, we would have had to sweat a lot. We would have had to sweat so much that we'd get dehydrated. So we wouldn't have survived in the present day heat that we are having here in Lahore or the heat in June. So high latent heat of vaporization has a larger cooling effect. Why? Because removal of large quantities of heat energy by evaporation. So the plant or the leaves, when they heat up and the light is absorbed for photosynthesis, Now that would be, it will heat up the plant. So it reduces, if it doesn't heat up, then that means proteins or enzymes will not be denatured. Also, it will reduce the rate of water loss by transpiration or the rate of water loss by evaporation at high temperatures. So you have to understand high latent heat of vaporization. So please remember that you need to first do the paper, which was the unsolved paper, which the video I am putting up on my channel. And then, of course, you need to look at this and try to do it yourself, look at your wordings, and then see where you wrote wrongly and how you can improve that. So high latent heat of vaporization results in large cooling effect. Plant leaves heat up and light absorbed by photosynthesis. And as the plant will cool down, so this will reduce proteins or enzymes being denatured. And it will also reduce the rate of water loss by transpiration at high temperatures. Just as I gave you an example that when this room is very hot and I'm sweating, well, the sweat must evaporate to cool me down. But if I had to sweat profusely, then I would have got dehydrated because it had a low latent heat of vaporization. Now, coming to question number 4, tuberculosis, TB, influenza, and polio are examples of infectious diseases. That means you can get it, they're infective, they're transmissible, they can be transmitted from one person to another. Explain what is meant by an infectious disease. Now, in order to get those 2 marks, you had to say caused by pathogens. Is transmissible. Because why couldn't you have said that? Because that's the word given in the question. The question says, what is an infectious disease? And you say infectious disease is a disease which is infectious. Well, then you must be a pretty changada. Because, I mean, if you're writing the words of the question, I'm saying, what is phloem sieve tubes? And you're saying, Miss, phloem sieve tubes are phloem sieve tubes. You're not telling me anything about that. You're just repeating what I said. So if you say, explain what is meant by infectious disease, and you say it is an infectious disease because those which cause and if you say it's an infectious disease or it's a disease which can infect others, even that is wrong because again you're using the word of the question. So the first thing you had to say was caused by a pathogen. You see, if you had scurvy, that is because of deficiency of vitamin C. That is not infectious because if you are not taking vitamin C, well, you have scurvy. How can I get it from you? Or how can you give it to somebody else, even if they drink in the same plate or the glass, drink in the same glass, or use your same fork and spoon which you have used? Well, that's not good. You're not going to get it. So, caused by a pathogen, and you could have said is transmissible. Now, or you could have said something else. You could have said it's contagious because here it says AW. AW means you could have said any alternate words, which means that you could have said a word which means the same thing. Now, if this is not written here, it wasn't written here. So you had to use the word pathogen. You couldn't have used caused by bacteria or caused by virus. No, pathogen, infectious disease could be anything. Could be caused by fungus, could be caused by plasmodium, which is a protoctist. So caused by pathogen. Now that's a typical term. You cannot, it didn't say AW there, it said AW here, which means is transmissible. You could have used other words like I just said, contagious. Now it says name a species of organism that cause TB. Now that of course is the whole name, Mycobacterium tuberculosis. Now here you could not have said prokaryote or you could not have said bacteria. Mycobacterium tuberculosis or you could have said Mycobacterium bovis. Or you could have said Mycobacterium bovis. Then it says immunity can be described as artificial or natural and passive or active. Artificial or natural, passive, active. Name the type of immunity that the mother gives to her baby through breast milk. What is that? Natural and passive. There's only one mark. But you have to know natural because there's nothing interference by anybody. And because the mother has suffered from a disease and she is passing on the antibodies to her fetus or to the either across the placenta or in breast milk. So she is giving ready-made antibodies. Now that is natural and passive. So You have to know this. I hope you can revise that. I have done this in my video. What is natural and what is all this natural and artificial and passive and active? Part C, the influenza virus can mutate frequently to produce different strains of the virus. A new vaccine is often necessary to stimulate the production of new antibodies to these new strains. Explain why different antibodies need to be produced to give immunity to these new strains. Now, first I'm going to explain this to you and then I'm going to give you the wordings of the mark scheme. So this is virus A and virus B, both cause influenza. It says can mutate to produce different strains of the virus. Now, in this situation, what we need to have antibodies like this. Now, in this situation, we have to have antibodies like this. So why do we need, they have different antigens. New vaccine contains an altered antigen. This is the antigen. This is the antigen here. This is the antigen here. So they're different antigens. Like this virus has this antigen. Another virus has this antigen. Antigen means surface markings. So new vaccine will contain the altered antigen. Antibodies are specific to antigens. So they are complementary shape. Then there's the variable region at the ends. So the variable regions at the ends are going to be different. As the influenza virus mutates, the antigens protein are altered. Now these will require antibodies with the different variable regions to be effective. Now let's look at the wording. New vaccine contains antigen of new strain. Antibodies are specific to antigen. Complementary to the antigen, the variable region is complementary to the antigen. Variable region is at the ends. As virus mutates, antigen protein altered, requiring antibodies with a different variable region. Now coming to the D part of the question, polio is a serious viral disease affecting young children. In 1996, polio caused paralysis in more than 75,000 children across Africa. A long-term vaccination program allowed the World Health Organization to declare that Africa was largely free of polio in 2020. Explain how vaccination programs can help to control the spread of infectious diseases such as polio. They've given you information that's a viral disease. You better be looking at it. So if this is the virus which causes polio, then what do they do in the vaccination? They give this weakened polio virus. So vaccination programs aim to vaccinate whole populations. Vaccine allows the children to have an immune response against the antigen. So this is the virus which causes polio, and we give this weakened virus. So the B lymphocytes will be selected, and memory cells will develop, and plasma cells will develop, and they will produce antibodies. But basically, we want the memory B cells. Why? Because the memory B cells, when the actual polio virus will enter, the memory B cells will change into plasma B cells and will make antibodies and will destroy the active live polio virus. So what we do is we give them weakened polio virus so that the B lymphocytes are selected and they are cloned and they differentiate into plasma B cells and memory B cells. But basically we want the memory B cells because when the person actually, the real live polio virus is going to invade the body, then he or she is not going to get polio. So he or she will not get destruction of the motor neuron, which goes to the muscles, leg muscles. And that's why people who've had polio will limp and they will be disabled for the rest of their life. You can't do much about it. You can't do anything after the person has had polio and the motor neuron has been destroyed. So vaccination programs aim to vaccinate whole populations. Then vaccine allows children to have an immune response. Memory B cells develop against polio. These children cannot transmit polio or infect others, so you break the transmission cycle. Then the unvaccinated children will of course will not get it. So it says unvaccinated non-responding children are protected, shielded by a large number of immunized children, which is called the herd immunity. So herd immunity means that you vaccinated 100 people and only 95 actually developed the immunity. So that is having 95 people are immune. That means it won't spread to other people. Then coming to the next part of the question. Now, antibiotics such as penicillin do not help to prevent the spread of viral diseases such as polio. Explain why penicillin is not effective against viruses. Why? Penicillin only acts against bacteria. Virus does not have any cell wall. Virus does not have any enzymes that are actually destroyed by antibiotics. Virus does not grow on growing cells, and virus does not have a cellular structure. So antibiotics actually, how do they act on bacteria? They destroy the cell wall, they act on the enzymes, so the enzymes cannot work. They act on the cell membrane, but virus does not have a cell membrane. So the wordings for this is vaccination program aim to vaccinate whole populations. A vaccine allows children to have the immune response. They develop memory B cells against polio. These children cannot transmit polio. Unvaccinated protected by large numbers of immunized children. And this achieves herd immunity. Just Google herd immunity and you'll know what that means. So the fact that the antibiotic penicillin does not work on viruses, because how does it kill bacteria? It actually disables an enzyme which is helpful in making the peptidoglycan cell wall. So penicillin only acts on bacteria. Virus does not have a cell wall. Virus does not have the enzyme that is inhibited by penicillin. And virus is acellular. It is in fact acellular. This is called virus particle. So any one of these and you got your 1 out of 1. So this was in sort of an easy question which checks whether you understood this part of the syllabus, which is antibiotics, how they work and why they do not work at viruses. In this second video, we have solved question 3 and 4, and another video will follow, which is question 5 and 6.